Good afternoon, or good morning, or good evening. We are running a very international presentation today. Welcome everyone to the NOAA Central Library. My name is Katie and I will be the library host for this presentation today. You are an attendee, so you are muted. And if you have questions for our speakers today, you will have to place those into the question or the chat panels, whichever one you see. If you are having any technical issues, like you cannot see the slides or you can't hear us, please try logging off of GoToWebinar and back in through your join link. That solves most issues. Please note that we are recording this, and any questions you ask or comments you have will be passed on to the speakers after the fact. And with that, I'm going to turn it over to Terry Rolls, who is going to introduce our very exciting topic and speakers.
Good afternoon. We're going to be having a very exciting talk about cancer in California sea lions, and have a panel of 4 scientists who have been working on this issue for a while. Dr. Frances Gulland is a veterinarian and research associate at the University of California, Davis School of Veterinary Medicine. She has worked at the Marine Mammal Center in California for 25 years, and she has chaired the U.S. Working Group on Marine Mammal Unusual Mortality Events, the Southern Sea Otter Recovery Implementation Team, and currently serves as a commissioner on the U.S. Marine Mammal Commission. Professor Ailsa Hall is currently emeritus professor at the University of St. Andrews Sea Mammal Research Unit, and she has been involved in a wide range of marine mammal epidemiological, toxicological, and physiological studies with collaborators from across the globe. Dr. Alissa Deming is the Director of Clinical Medicine at the Pacific Marine Mammal Center. Her principal interest in her work has been in studying disease patterns in marine mammal populations to better understand the impacts of human and environmental factors on ecosystem health. And the final panel member is Dr. Irvin Schultz, who is a program manager of the Environmental Chemistry Group at NOAA's Northwest Fisheries Science Center. His area of expertise includes analytical chemistry, in vitro and in vivo extrapolations, toxicokinetics, and biological modeling. Dr. Gulland, I'll turn it over to you.
Thank you, Terry. Well, good morning, everyone. Before I start, I'd like to say that although you're hearing from 3 of us today, the work on cancer in California sea lions has been that of many. And in days of yore, when we used to travel and get together to talk about research, being together sparked the idea of creating a sea lion cancer consortium so that we could freely collaborate and share information ideas. So all the people listed on this slide here have contributed to the work that we'll be talking about today. Next slide, please. So California sea lions are an abundant population that range along the west coast of the United States, from Baja California in the south up to British Columbia in the north. They breed on Channel Island, on the islands off the central California coast in June and July. And after the breeding season, the males migrate north to feed. They actually don't feed during the breeding season, whereas the females stay close to the islands to nurse their pups for up to a year. And it's these females upon which the study is based, because during the year they strand along the California coast for a variety of reasons. Next slide, please. When these animals strand and they're brought to rehabilitation centers, animals that die are examined at post-mortem to determine the cause of death. And over the last 25 years, between 17 and 22% of stranded adult sea lions that are examined at post-mortem have cancer, which is a urogenital carcinoma that originates in the cervix in females and the penis in males. And these are animals that die due to the spread of this cancer from these original early lesions in the reproductive tract to a disseminated cancer. And over the years, the prevalence of cancer, meaning the number of animals with cancer out of the number of animals examined, has remained roughly constant, although the number of sea lions stranding has increased with population increases. and other causes of stranding, such as poisoning with domoic acid, have also increased. Next slide, please. Here on the left, you see photographs of how the animals look before they're examined at post-mortem, and there's swelling of the hind end and the perineal area due to, as you see on the right, these massive tumors that obstruct the ureters, the lymphatics, and the blood drainage from the hind flippers and the perineal area. Next. When the animals die or are euthanized for humane reasons because their prognosis is poor, the cancers have metastasized throughout the body. And here you can see lesions scattered along the mesentery and all over the liver. And I guess we should have had a spoiler alert. I hope no one's having their lunch while they watch these slides. Next, please. These are the early lesions. The above picture is the penile plaque on the males, and below you see a cervical mass in the females. They start as these raised nodular lesions and then spread very quickly to adjacent lymph nodes and then throughout the body. Next. Early histological examination of these lesions in the late 1990s revealed that in the vaginal tissue, there were quite clear intranuclear inclusions, and these were also observed in the penile lesions of males. Next slide, please. So early research into the cause of these inclusions, the characteristics of them, electron microscopy revealed herpes-like particles there on the left. And then PCR revealed sequences of a herpes virus that was a novel herpes virus, not previously described. So we called it uterine herpes virus 1. And it was closely related to sequences that were closely related to human herpes virus 8, which is also a gamma herpes virus and is associated with Kaposi's sarcoma in humans, a cancer. Next slide, please. So given that we could see these herpes inclusions in many of the feline cancers, the obvious question was, how was it associated with the carcinoma? So to investigate this, we collected tissues from stranded animals, both with and without cancer, and then sections of these tissues were examined by PCR for a DNA polymerase gene specific to the uterine herpes virus 1. And this early work was done by Don King, pictured on the right here at the University of California, Davis in Jeff Stott's lab. And we found that in samples of about 20 to 30 animals, the prevalence was higher in cancer tissues, but we also found it in non-cancer tissues. Next. This graph shows the results of this work that was published by Beth Buckles as part of her PhD work in the early 2000s in Dr. Lowenstein's lab at the University of California, Davis. And what we're looking at here are tissues from animals with cancer in red and tissues from animals in blue without cancer. And the left is the top end of the sea lion, the right is the bottom end, so trachea, salivary gland, etc., through to ovary, uterus, cervix, and vagina on the right-hand end. And the main point of this slide is to show you that although many of these tissues did have cancer tissues in red, had uterine herpes virus, there was also virus in some tissues from animals without cancer, namely salivary gland and uterus in a few animals in the controls. Next slide, please. And to further investigate the distribution of this virus, we collaborated with Bob DeLong and Sharon Meline from the Mammal lab on their long-term studies on San Miguel Island in California. And 250 California sea lions that were caught for health examinations had swabs collected from the urogenital area and from the pharynx. PCR, again, for the same DNA polymerase gene of the uterine herpes virus 1 was used, next slide, to screen these swabs. Next. And in these swabs, we found that if we look at the vagina and prepuce first on the left, the prevalence of uterine herpes virus 1 DNA increased with age and was higher in males. You can see 43% of adult males compared to 23% of adult females had the DNA. And then looking at the pharynx, very few samples were positive for virus. Only one adult male had a positive pharyngeal sample. So these results basically indicate that the herpes virus is present in healthy wild sea lions. It's more common in the reproductive tract and twice as common in males and females. So this suggests that it's sexually transmitted as males mate with multiple females and therefore are more likely to have a higher sexually transmitted disease. So this led us to several questions. What really is the role of this herpes virus in the sea lion cancer? Is it causative? Does infection with the virus cause cells to transform? Or is it simply a sexually transmitted subclinical disease that replicates in the rapidly dividing cancer cells of the reproductive tract? And we do know that in humans, there are a number of cofactors involved with cancers that do have a viral etiology. And some of these are listed here, such as concurrent infection with immunosuppressive viruses or chlamydia, chronic inflammation, genetic susceptibility, and then exposure to some toxins, especially the PCBs and diethylstilbestrol. So in the marine mammal world, we're always concerned about persistent organic chlorines in these animals because they feed high in the trophic system. And especially California sea lions, we know that they're feeding in the Southern California bight, an area that's heavily contaminated with DDTs and also PCBs. So some earlier work led by Gina Italo at the Northwest Fisheries Science Center, where Irvin now works after Gina's retirement, showed that these a suite of organochlorines, a number of different PCBs, DDTs, and hexachlorobenzene were high in blubber samples collected from sea lions with cancer. And in this figure, we're looking at sea lions with cancer circled in red and sea lions without cancer in yellow. And the bars are showing the mean PCB levels, mean of the log sum PCB levels in the animals, either red or yellow groups, cancer or non-cancer animals. What we're doing is we're looking at some PCBs expressed either per lipid weight or per wet weight of blubber. And either way we look at it, animals with cancer have higher levels than animals without. Next slide, please. However, we were concerned immediately. We're looking at the results of this study because animals with cancer are usually very thin, having been chronically sick, and lipid is lost from the blubber. Whereas the animals that we used as controls were stranded animals that had died from mostly gunshot injuries, so had died acutely, were fat, and had thick blubber levels. So to address this concern, we took advantage of California sea lions that were stranding with domoic acid toxicosis. And as the animals came in, they were fat, acutely poisoned with domoic acid. But as they went through their rehabilitation, their blubber lipid levels changed. When they first were admitted to rehabilitation, they didn't feed for 2 to 3 weeks. And then once they recovered from the toxicosis, they began to feed again. So over this period, over the first 20 days, look at the graph on the left, these animals were losing weight, and then they regained weight, and we look at the period on the right of mass gain. And what we did is we took blubber biopsies from these animals at admission to rehabilitation at the nadir of their weight loss just before they started to feed again, or as they began to feed again, and then a third blubber sample at release. And these samples were examined. Total PCB levels were determined at the, again, Northwest Fisheries Science Center. And you can see that during the period of mass loss, the concentration of PCBs per lipid weight increased in blubber samples. And then over the period of mass gain, the blubber PCB concentrations decreased. So there is certainly an effect of blubber, lipid percent, and animals' body condition on PCB levels. Next slide, please. So this kept us thinking about, well, what really is the role of PCBs? Are we simply getting higher levels in animals with cancer because they're losing weight? And we wanted to understand more about how sea lions are exposed to PCBs and DDTs over their lifetime. So again, taking advantage of sea lions that stranded and died from domoic acid poisoning, many of these animals were pregnant, and so we could collect paired blubber samples from mother-pup pairs in late gestation. And these data are summarized by Denise Gregg in a paper published in 2007. And the slide on the right, the graph on the right, shows you the relationship between specific PCB congeners in mum and pup. And the essential message from these samples from these 20 animals is that there is transfer of PCBs across the placenta, and thus prenatal exposure of developing California sea lion pups to PCBs while their reproductive tracts are developing. Next slide, please. Concurrently to all this work, Karina Acevedo was working on her PhD, looking at the associations between inbreeding depression and different causes of death in animals that stranded. And she found that animals that had carcinoma were significantly more inbred than animals dying from other causes. In closer examination of these data, she realized that most of this association was resulting from a genotype at the PV11 microsatellite, a specific microsatellite among the suite of microsatellites that were used to look at inbreeding depression. So in some follow-up work by Helen Browning for her PhD, she found that this PV11 microsatellite was located within the heparinase 2 gene, and indeed, specific type of this microsatellite was associated with the risk of developing cancer. Next slide, please. So now we have sort of three skeins to this cancer pathogenesis. We appear to have a virus that's associated with the cancer, but also present in healthy animals. We have high levels of PCB and prenatal exposure. And we also have some genetic associations with the cancer. And we know from work in laboratory animals that carcinogenesis is multifactorial. And for a normal cell to become a carcinoma cell, there has to be an initiating event, a first mutation, and then promotion and fixation of that DNA damage for the cell to become a replicating carcinoma cell. So this led us to propose that carcinogenesis in these sea lions is multifactorial, and what we really needed was a study that examined the three factors that we'd identified to date in a large sample size to look at their relative importance, rather than these discrete studies on 20 to 30 animals at a time. Next slide, please. So this led us to develop a case control study. And at this point, I'm going to hand over to Dr. Ailsa Hall, the epidemiologist who really led the study design, and talk us through how we examine these multiple factors in one study.
Thank you, Frances. So for those of you not familiar with case control studies, this is one of the classical epidemiological study designs that was pioneered really by Dole and Hill in the 1950s and 60s with their smoking and lung cancer risk studies. And in this design, the subjects are selected according to their disease status and then further classified according to their exposure status. And by exposure, we mean the broad range of those factors that that Frances has identified. Next. So if this exposure variable is what we call dichotomous, so if it's a factor that you can split into just exposed and unexposed, then the simplest analysis of these data yields what we call a cross-product ratio. Next. And in this little table, you can see cases and controls, and then you split them into those exposed with the factor or without the factor. Next. And it gives you an odds ratio. So this cross-product ratio, A times D over B times C. Next. So the odds ratio is the effect measure that you get from a case control study, and it's equivalent to the odds of being exposed amongst the cases divided by the odds of being exposed amongst the controls. And those of you that have bet on the horses have a good idea of what an odds is and how it relates to probability and chance and the likelihood of a particular outcome. It is a measure of average risk, but we have to be a little bit careful with the term risk because it's not equivalent to a relative risk that you would get from, say, a cohort study, because that only really works when the outcome of interest is quite rare. But it can be converted into a probability. And so we're looking at probability of disease in an individual in a specified time period, estimated by this odds ratio being greater than 1. So if there's no difference between your cases and controls, then you have an odds ratio of unity. Next. And of course, it's really important in any of these epidemiological studies to be robust about your case and control selection. So cases and controls are randomly selected from a defined source population. In our case, the California sea lions that breed on the Channel Islands and come through the Marine Mammal Centre. Next. And the controls should be selected from that same population that gave rise to the cases, and most importantly, independent of their exposure status. So the pathway by which animals become a control is the same pathway by which animals become a case. And so the time during which that individual is eligible is also important. So the eligibility must be the same between the cases and the controls. So they would, animals that were identified as controls would be a case if the disease had developed in those individuals. Next, please. It's a little just diagram to summarise that. So on the right, we have the cases and controls in the onset of our study, and then we're looking back in time as our direction of inquiry into exposed and unexposed. And the hypothesis is that there's higher exposure in the cases than the controls and vice versa. Next. What's obviously important, as Francis pointed out with the blubber thickness aspect of the contaminants, is that other factors that could be responsible for differences in disease frequency between exposed and unexposed must be controlled for when looking at these odds ratios. Next. We were able to do that and estimate the odds ratios and probabilities using a logistic regression approach, which is a standard approach for case control study analysis, and that allows for effects of confounders to be accounted for in the subsequent analysis. So our cases were the female sea lions that stranded along California coast from St. Louis Obispo to Humboldt counties between 1998 and 2017. That were diagnosed as having UGC on examination at the Marine Mammal Centre, which was 179 animals. Controls were the same, but those that didn't have UGC, mostly animals that died of domoic acid toxicosis or trauma, had about the same number of cases as controls, and you can see very, very great improvement in the sample size there. And you really need more controls than you have cases to get a good estimate of the odds ratio. So the first factor we looked at was the genetic basis, and you can see here the results from Helen's study. It was quite weak evidence that there was a link between homozygosity at PV11 and the development of UGC. Next slide, please. And then in our study, when we looked at that again in the same way, using the same microsatellite markers, we found that actually that finding was not sustained, and the odds ratio, the confidence interval there, as you can see, spans the value of 1, and therefore that's not really a significant finding. However, the herpes status was highly significant, as you saw in the bottom of that slide there. Yeah, thanks. So that was a 31 at the mean estimate for the odds ratio, and we had only 7 cases that were negative for herpes virus on the PCR. Next slide. So the contaminants were the other really important factor in this study, and Gina measured 5 different classes which all correlated positively with each other in terms of concentrations in the blubber. Next. So what we did was looked at those distributions of the concentrations you can see here between the cases and the controls. So the cases are above the line in red and the controls below the line in blue. And we summed all of those contaminant concentrations together on the same scale. So in the bottom right is that figure with the sum of the blubber contaminants, which was the variable that we used in the analysis in the end. And you can see even visually that there's a slight difference in the distribution, in that the control concentrations are skewed towards the lower end compared to the concentrations in the cases. Next slide, please. And of course, we have this negative relationship between blubber contaminant concentrations and blubber depth, so where they concentrate in the remaining lipid as the animals lose mass. Next slide. So we had to control for that, but here's a summary then for the herpes status and the distribution of contaminants. And on the left, you can see we were pretty sparse in cases without herpes virus. And on the right, you can see the herpes positive distribution, again, with those controls having slightly lower contaminant concentrations. Next slide, please. So in the final model, the blubber depth was controlled for, and to visualize that, I've plotted here 3 blubber depths and the relationships between the contaminants and the herpes and the probability of cancer for thin blubbers at the top at 0.5 centimetre depth, a mean blubber of 1.1, which was the mean for the whole population, and a thicker blubber depth of 2 centimetres. And you can see the probability of being a cancer case increases with blubber contaminant concentrations. However, that likelihood is considerably higher for herpes positive animals across all the blubber depths. And so herpes is clearly the main driver of this disease, combined with an increased risk with the contaminants. And so we needed a good understanding of the mechanism of that. And that's where I can hand over to Alissa, and she can tell you about her research on the herpes.
Okay, perfect. Thanks, Ailsa. Okay, so now we're going to talk a little bit more about how we more definitively determined that this herpes virus was actually causative and not correlative in the disease with the sea lions. And that can be a little bit tricky because herpes viruses are actually quite prevalent in a lot of hosts that they infect. So if we look at people, almost 100% of the people by the time they're adults are infected with at least one herpes virus, news that you guys probably don't want to hear. But there's actually 8 herpes viruses that infect people. So oftentimes people think of cold sores or genital lesions with herpes viruses, but there's also chickenpox, which is a herpes virus, mononucleosis or kissing disease or mono that teenagers typically catch in high school. That's Epstein-Barr, that's a herpes virus. Kaposi sarcoma, as Francis mentioned earlier, which those later 2 can be oncogenic herpes viruses or cause or be associated with cancer. The interesting thing about those 2 oncogenic human herpes viruses is it doesn't cause cancer in all of the people that it infects. In fact, it only causes cancer in a very small subset of people. So researchers have found that there has to be some kind of triggering event, and oftentimes that has something to do with a malfunction of the immune system or some kind of immune suppression that can be related or caused by a number of things that causes the herpes virus to go from a more dormant or a quiet state in cells, because you're infected for a lifetime once you're infected, but typically the herpes virus is laying low and the immune system is keeping it in check. But if your immune system is compromised for some reason, the herpes virus can come out of dormancy and start causing disease. A good example of that is people that have chickenpox when they're younger. When your immune system gets suppressed a little bit as you get older, you can break out with shingles. Cancer is also another thing that significantly impacts people. If you're a woman, you have a 1 in 3 chance of getting cancer in your lifetime, and if you're a man, you have a 1 in 2 chance. Luckily, with screenings and treatment options, a lot of times there's ways to treat those cancers, but something a lot of people don't realize is over 20% of cancers are associated with infectious agents. So if we can do things like preventing the infection, the chickenpox vaccine, or trying to keep people from getting things like human papillomavirus and developing a vaccine for that, we can really impact the prevalence of cancer. Additionally, if we can figure out the mechanisms behind these cancers being triggered to go from a dormant state to something that can cause disease, is also a way that we can significantly decrease these 20% of cancers that are known to be associated with infectious agents. So what does that have to do with sea lions and the animals that we see? Well, the fact that we've seen so many sea lions that have cancer have a sexually transmitted genital herpes virus raises a lot of flags that this is a virally associated disease. But it kind of throws a wrench in the fact that there are several animals that never develop the cancer that test positive for this.
So this next section of the talk is going to discuss some ways that we tried to determine whether or not this herpes virus was causative or correlated. So the first thing we did is we sequenced and annotated the OTHV1 genome. We did this using next generation sequencing. MiSeq and PacBio were the two main platforms we used, and these are technologies that are very rapidly advancing. So this is work that could not have been done 10 years ago for the same price point and rapidness that it was done. And this allowed us to get a draft genome of almost 150 kilobase pairs of otorine herpes virus 1. And then we annotated that genome and looked for genes that are in other herpes viruses that are known to give it characteristics of oncogenicity or ability to cause cancer. The next thing we did is we wanted to look at viral loads. So we know that once you're infected with a herpes virus, some herpes viruses stay with you forever and typically stay latent in your system, but it's when those herpes viruses recrudesce that you develop issues. So looking at quantitative values by using qPCR of how much herpes virus an animal has can help us determine if that herpes virus infection is just present but not being actively expressed, or if that herpes virus is being highly expressed. And then the last thing we did is we looked specifically at the viral expression, and this was probably one of the strongest clues or evidence that the herpes virus is associated with the cancer itself. So this was also a very new technology that we developed. Dr. Katie Colgrove and Dr. Jennifer Luff really helped with portions of this part of the project. And basically, because we have the DNA sequence of this herpes virus, we can figure out what the mRNA sequence is. And then looking at the mRNA sequence, that proves that the herpes virus genes that we're specifically looking at, that we designed these in situ hybridization probes to, is actively being expressed. And then by looking at that under the microscope, you can actually see where that herpes virus is being expressed within the architecture of the tissue or cervix itself. So just very quickly, this is a phylogenetic tree of a variety of herpes viruses, alpha, beta, and gamma, and various hosts that they infect. We did also sequence another pinniped virus called otorine herpes virus 1 that infects northern fur seals, but has not been associated with causing cancer in them. And we wanted to use that as a comparison to see what an oncogenic pinniped herpes virus looked like versus a non-oncogenic one. And interestingly, when we ran our phylogenetic analysis of this, those two herpes viruses came together, which is to be expected, but they also grouped with a dolphinid herpes virus that causes genital lesions in dolphins. And they branched very early on from the gamma herpes virus. And this early branching is a novel genus associated with the herpes virus that we're proposing to call marmavirus. So next up, we wanted to look at the viral loads in cervical tissue. So we PCR tested control and cancer cases. 163 of the control cases were tested, and 36% of those tested positive. And then all of the cancer cases that we had in this subset of animals tested positive for OTHV1. Then we did qPCR on all of the animals that tested positive for cancer, or for OTHV1. So the control animals that did not have cancer but did have herpes, the concentration or the load of the virus was only about 10,000 copies of OTHV1 per nanogram of DNA. Whereas in the cancer animals, it was exponentially higher, where they had a lot more herpes in their samples, and they had about 10 million OTHV1 copies per nanogram of DNA. So that's already our first clue that these cancer animals have a lot more herpes virus actively being expressed compared to the control animals that are infected but don't have cancer. Next, we wanted to know the probability of having cancer. So on this top line, your x-axis is the concentration or the viral load of OTHV1 in cancer or control animals. And the top line is the concentration of viral load in cancer animals. The bottom line is the concentration of viral load in non-cancer animals. And you can use this model now once, if you test a sea lion cervix biopsy for OTHV1 using qPCR to determine the probability of having an animal with cancer. So if an animal has a low viral load and is further to the left, you can follow this line where it hits and say it has less than a 20% chance of having urogenital carcinoma. As the viral load goes up, the chances of a sea lion having urogenital carcinoma also goes up. And then lastly, we're going to go over some histologic lesions of California sea lion cervix. This actually looks quite similar, I'm told, to what a human cervix would look like. And you can see there's those top purple cells, or the top layer of cells is the cervical epithelium, pseudostratified epithelial layer on the cervix. And then just below that is the stroma or connective tissue underlying the surface of the cervix. Now, cells have to follow rules, and typically they are good neighbors. So they don't invade in spaces where they shouldn't be. They replicate only when they're told, and they go through apoptosis or dying when they're supposed to. When cancer starts to develop, you can see on the left of this histologic image, there's normal cervical epithelium. And then towards the right of this image, the normal cervical epithelium starts diving down deeply into the underlying stroma, not following those rules that it should be following based off of a good neighbor cell. And this is kind of the definition of what cancer is. So remember this picture, we'll come to it in a few more slides again. But we wanted to know if this animal is infected with herpes, is the herpes being actively expressed kind of uniformly across in the normal epithelial cells as well as this carcinoma in situ, the cancer starting, or is it preferentially in one area of this slide? So as the cancer progresses, it's not following the rules even more, kind of spreading deeper into the underlying stroma, but still not becoming invasive yet. So this is still an early on carcinoma in situ urogenital carcinoma in the cervix of a California sea lion. And then when it becomes invasive, you just kind of can't recognize it at all. And it breaks off and goes either into the underlying stroma tissue, or also gets spread in the blood vessels where the cells evolve to break off and go into the blood vessels, metastasize to different sites, and evolve to be able to settle down in those distant sites. And using sea lions to better understand that process or that evolution of cancer from going from a primary mass in a localized site to a more distant site and metastasizing is a very important area of research, because if you can stop that metastatic spread, you can stop the majority of the mortality that you see with any type of cancer. So this is that slide that we saw earlier where it was the very early carcinoma in situ, and you can see the normal cells to the left, and then the early carcinoma in situ diving down into the underlying stroma. So the pink dots that you guys see that are localized only in the carcinoma in situ part of the tissue are the mRNA OTHV1 probes that we used. So this is just one probe looking at one gene in the genome of the herpes virus that we sequenced. And it's looking specifically at messenger RNA. So that means this is a lot of binding in an in situ hybridization slide. So this is showing us that the herpes virus is not only very highly expressed, but it's exclusively expressed in the cancer tissue, but not in the surrounding healthy epithelium. So we looked at this, we looked at 5 different odorine herpes virus 1 genes, 3 of which we thought could be suspect oncogenes because they're homologous to other known herpes viruses that these genes cause cancer in. And we looked at animals that were healthy, which is the panel to the left. The panel in the middle is the carcinoma in situ, and the panel on the right is the invasive. And then we have a positive control and a negative control. And as you can see in all of the animals that we looked at, there was no significant expression of the herpes virus in the healthy animals, no matter the gene that we looked at, except for the positive control. There was expression in that. The next column in the middle is the carcinoma in situ animals, and you can see that it is exclusively expressed, the herpes virus, only within that tumor tissue, but nowhere else. And then the same pattern was seen in the invasive tissue. And this was consistent with all animals that we looked at. So we feel like, you know, short of infecting sea lions with herpes viruses and stressing them out and then giving them contaminants and seeing if they develop cancer, these findings between the high viral loads, the high viral expression, and the viral expression only being expressed within the cancer tissue itself and not the surrounding tissue, and not being expressed in the healthy animals that are infected, is supportive of the fact that this herpes virus is causative. So just some take-home messages from all of this is this cancer has an incredibly high prevalence for any cancer in any mammals. Typically, you don't see the same type of cancer at this level of prevalence in any mammal population. So that's remarkable in and of itself. The herpes virus, both the cancer control paper and the herpes virus paper are strongly supportive that herpes is required for the cancer to happen, but it is not the only thing that has to happen. So that's when things like contaminants come into play, or other stressors that could potentially induce the transformation of a latent or dormant herpes virus into one that's going to develop into a malignancy. Next, it's a really important message. The prospective sampling that happened over 25 years at the Marine Mammal Center, that Frances had the foresight to start collecting these samples even before she knew what she was going to do with them, filled the freezers at the Marine Mammal Center with lots of samples from California sea lions, both with and without cancer. And all of these animals were used in the cancer control study. So we now have contaminant levels for these animals, we have herpes virus status for these animals, and then the Marine Mammal Center has a ton of tissue from these animals that future research can be done. So we can really now be positioned to start using sea lions to study a naturally occurring disease model in better understanding what causes viral carcinogenesis, as well as better understanding the metastatic processes. And there's a lot to be said. There's pluses and minuses for studying a disease in a natural setting. If you're not in a lab, you can't control the parameters, and it can sometimes be a little bit challenging to tease apart all of the different influences that can be impacting the development of cancer. However, that's how cancer develops in the real world. So studying sea lions that have all of these external stressors and these multifactorial components that are causing the development of a multi-step process leading from herpes virus infection to cancer more truly represents how cancer develops in people. So we propose that these sea lions are going to be an excellent model to better understand California sea lion cancer, but also bigger picture-wise, better understand cancer in all kinds of animals, in people, and better understanding metastatic disease processes. So if you have come up with brilliant projects and things that you think that we would be interested about when it comes to your insight in what's causing California sea lion cancer, what these samples can be used for in the future, you can visit this website. There's a little form in the bottom you can fill out and let us know your email address and your interest, and we can kind of link you into our Sea Lion Cancer Consortium group and really encourage collaborative effort and using this massive bank of samples that are at the Marine Mammal Center that Frances helped pull together to help better study cancer in California sea lions, metastatic disease, and virally induced cancers. So with that, I will just say thank you. Like Frances said in the opening, there were a lot of people that helped contribute to this over the past 20, 25 years. NOAA Northwest Fisheries for running a lot of our contaminant samples, and then of course the Stranding Network and the NIMS Marine Mammal Health Program for allowing us to do this work, as well as the Marine Mammal Center volunteers take fabulous care of these animals, and without them we wouldn't be able to do projects like this. The Jeffrey Hughes Research Fellowship, and of course the Sea Lions that helped us do this work. And with that, we will take questions, I think.
Awesome. Thank you, guys. This was a very enlightening and very interesting talk for someone who doesn't have any clue about any of this. So thank you. As a reminder, everyone, this is Katie, and I am taking your questions. So please place those into the question or chat panel. We do have our first question from Terry Rolls. Have you observed urogenital cancer in other otarids?
No, great question. No, we haven't. And we're also curious about the source or the origin of this herpes virus. So although we haven't seen carcinoma in northern fur seals, we have sort of explored their herpes viruses, and maybe Alissa, you might want to speak to that.
Yeah, herpes viruses typically co-evolve with their hosts, so they don't jump species very easily, which is good when it comes to the risk of transmission to non-California sea lion species. But I think there is potential risk for a species jump if the northern fur seals decide to commingle with the California sea lions, which on occasion occurs in a sexual manner. But it hasn't been documented, no, not yet.
Thank you. I'm going to move on to our next question. Are there any older tissue samples that indicate approximately when OTHV started to cause cancer in California sea lions?
I think it was first documented in 1979 and has been seen pretty consistently within the population along the California coast over the past several decades. And it is likely well before the time that people would have been collecting samples that this disease process probably occurred. But with the component of the contaminants and the high contaminant burden that has been distributed along the West Coast where the sea lions have rookeries, it's likely that that contribution may have worsened the prevalence of the cancer in the California sea lions.
Thanks, Alissa. Francis wants to ask, can you speak a little bit about the current exposure to PCBs and DDTs? Sorry, PCBs and DDTs.
Sorry, speak to it. Well, it's still happening. I'm not quite sure sort of how to respond to that. Maybe you might. There's certainly ongoing exposure.
We haven't seen in that whole time series, we haven't seen a decline. So from the 1998 sample through to 2017, we didn't see a decline in those contaminant levels very much. And that's probably because the legacy is from the female to the pup. So when you're born, you have a legacy of contaminants from your mother, which then follows you through to your offspring. So it's going to be a long time before those contaminant concentrations decline. So that risk is going to remain until some decades before those persistent pollutants have made their way out of the food chain.
So, Irv, are you on? Do you want to address?
I would just add also that those two classes of contaminants are still among the most abundant, highest concentrations we see in marine mammal tissues compared to other classes.
Great, thank you. Following up on that, how they acquire it, Ailsa, if they're acquiring this herpes virus from their mothers at birth, do the cancers generally remain dormant until sexual maturity?
Sorry, no, that was the contaminants they get from their mother, not the herpes virus.
Ah, yes, thank you for clarifying.
Okay.
Next up, has this type of research been conducted on captive animals who have been in captivity much of their life?
There has been some,
herpes is presumably sexually transmitted.
yeah, and there has been some screening of managed care animals, and there has been potentially some herpes virus within the managed population that is spread amongst animals. It's interesting because the managed care animals typically don't develop the urogenital carcinoma if they're infected until much later in their life. So the animals in managed care, you don't see the urogenital carcinoma until they're like 28, 30 years old, whereas the sea lions that are wild develop the cancer at a much younger age. So that is potentially where a component comes in where there's some kind of immune suppressive effect, potentially associated with the contaminants the wild sea lions are exposed to that help induce the process of carcinogenesis with the herpes virus infection. Whereas the animals in managed care aren't stressed, aren't eating food that have contaminants in it. And so it's the waning of the immune system at an older age that likely predisposes those older managed care animals to developing urogenital carcinoma.
The other feature about captive sea lions versus wild ones is that they do get a whole variety of cancers, whereas in the wild stranded sea lions, it's pretty much this urogenital carcinoma that we see. There are a few reports, a few observations of some other cancers, but 99% of them are urogenital carcinoma. Whereas if you look at the literature of cancers reported in captive sea lions, there are a variety of sarcomas, myelomas, lipomas, etc. So there's clearly, it is a multifactorial process, and we have factors in the wild that aren't affecting animals once they're under human care.
Great, thank you. Our next question, are you looking at multiple exposures in these animals?
I'm sorry, what kind of exposures?
The questioner said more modern chemical exposures. So I'm not sure what they mean by modern chemical, if they want to.
I think that's an area for future research. I think that there's a lot of stuff kind of coming into our ocean environment, especially in these marine mammal species that are so coastally associated. So I think there is room to do some exploratory work to see if there's any additional contaminants that could be contributing to the development of the disease, definitely.
Yeah, and I could add, sometimes when people think of modern chemicals, that it gets also considered in this other group called contaminants of emerging concern, or CECs, and that includes things like perfluorinated compounds and pharmaceuticals and treated sewage, among other things. And that is like an active area of research, but many of those aren't as bioaccumulative as the way PCBs and DDTs are. So exposure tends to be more associated with the discharge sites.
Thanks, Irv. I think this next question may be up your alley as well. Has it been established where the PCB contamination originated?
You can't really point to specific spots. There's many locations where PCBs were used, and they had decades-long use in many products. So there's many point and non-point sources for PCBs. And then because of their bioaccumulative and persistent nature, they've just distributed throughout many environments. So now it's not as easy to point to a particular source the way you might with other types of contaminants.
And I think to build on that too, California sea lions range along the West Coast, so that these females may have fed, they have fed anywhere really from the Southern California bight up to Northern California, sometimes even Oregon. So over their lifetime, they'll have picked up contaminants from prey from a number of sources.
Great, thank you. Their next question, has the immune response been characterized for the lymphocyte cell population during carcinoma in situ versus the period it wanes prior to metastasis, sorry, stumbling over words.
So comparing lymphocyte responsiveness from animals at different stages of cancer, no. So there has been quite a bit of work on lymphocyte function, mostly with Sylvain de Guise, Milton Levin, and Karina Acevedo. All of them have done quite a bit of work, but all the samples have been from single time points. So, you know, we get a stranded animal, it's at that time point of its cancer. Lymphocytes have been collected for immune function work, and we have data from those, but we haven't had the opportunity to sample a single animal longitudinally through stages of cancer. Once we know the cancer is fatal, these animals are euthanized for humane reasons.
Yeah, and I
So
wonder,
interesting.
I wonder if the person that asked that question is a pathologist, because in that early carcinoma in situ image that we showed, that histo image, you saw a lot of angry immune cells, lymphocytes and neutrophils, mostly lymphocytes, reacting to that carcinoma in situ. And it's very likely that the inflammatory state of the immune system reacting in a variety of cancers can kind of drive the progression of that cancer. So I think that that is also an area of future research, and that may hold a lot of answers. Just the immune environment and the microenvironment when these cancers are developing to help just better understand those basic mechanisms behind cellular transformation and malignancy over time.
Great, thank you. So our next question. So is the big takeaway idea that persistent contamination, primarily by PCBs, cause immunosuppression, allowing the herpes virus to spread? Is that the big takeaway?
I think that's one possible explanation, but we don't have the data to say that. And these animals aren't uniformly immunocompromised. It's not as if they're coming down with a suite of different secondary diseases. It's very specific to this urogenital carcinoma. So it may be that there's a specific part of the immune response to the herpes virus that is suppressed. But it's also likely that these prenatal exposure that have estrogenic effects are altering the receptor distribution in the reproductive tract. And we know there's some evidence from humans, exposed to diethylstilbestrol and estrogenic compounds that increases the likelihood of cancer in the reproductive tract later in life. So that's just a very different mechanistic explanation. But the reality is, so far we essentially have an epidemiological study, we have some mechanistic work on the herpes by Alissa, but the actual changes in immune response or the changes in the reproductive tract receptors that may happen, we don't know about yet.
Thanks, Francis. Next question. How frequent is this virus in these populations, and how frequently does it lead to cancer and mortality? How large of an effect can it have on a population?
Well, there's multiple questions. How frequent is the virus in the overall population? We really have the data from Beth Buckle's work that I showed the prevalence in 250 animals, where the virus was in 40% of males and 23% of adult females. So that's how common it is in a sample size of 250 apparently healthy animals. Whether it impacts mortality is something that we've been looking into, and Andy Dobson at Princeton has been doing some modeling. And it's basically difficult to get to, because the number of cancer animals we're seeing in the stranded population is high, but that's still a very small percentage of the overall sea lion population that's somewhere near 300,000. And there are such other dramatic effects on mortality from changes in oceanographic features like El Nino events that pretty much take out a cohort of pups. There is mortality from domoic acid toxicosis, there's mortality from leptospirosis. So the incremental mortality from cancer, at the moment, we don't have a good measure of that. And as far as overall impact on the population from the mortality, because the virus is sexually transmitted, most of these animals, once they die of cancer, will probably have already reproduced. So they're still generating a couple of kids before they die. So the preliminary modeling work that Andy has done doesn't show it as a major driver of population level mortality.
Okay, thank you. Our next question. Has any work been done to determine if mercury accumulation influences the prominence of herpes virus and cancer in these populations?
we haven't built mercury levels into the case control study design, but we meaning the people we collaborate with, notably Todd O'Hara's group and Laurie Rea in Alaska, they have done quite a bit of work on mercury in California sea lions, and they have mercury exposure. There is mercury, but we don't have any information on association with cancer.
Thanks, Francis. Alissa, I think you kind of introduced this idea. In general, is it likely that 100% of humans have not only a herpes virus, but also some level of PCBs in our bodies?
I think Irv's shaking his head yes, so I'm going to go with yes.
For sure. Everybody's exposed.
And I think that's the key to these sea lions is, they're top level predators, they're mammals, they have live birth, they nurse their young, they're exposed to similar contaminants that we're exposed to from the seafood that they eat, they have environmental stressors, and the development of cancer is typically never one thing. It's not just HPV causes cervical cancer in people, it's, potentially smoking or immune suppression for some other reason or other contaminant levels. So it is challenging to tease apart all of those multiple factors, and the cancer control paper and more definitively determining that this is an oncogenically induced cancer were just the first two steps to proving that this is going to be a great model to be using to help explore a lot of these other things that people are asking, like mercury and other components. So I think that there's a lot of work to be done, and I think they can really hold the key to better understanding the metastatic process as well as the virally induced oncogenic process by using these animals as a model.
I think a lot of these questions are great. So what I would like to take this opportunity to say is what really took the time is building up an archive of tissues from cases of cancer and age-matched controls, and just generating that tissue bank. You had to have histology to determine that an animal was clearly a control, age of the animal, measurements. So as Alissa said, these tissues are available, they're in the archive, and if there are ideas for pursuing other avenues, we'd love to take advantage of these samples, because it really took, it took 25 years to get this good, several hundred samples from known cases, known controls to build these questions on. So please do contact us.
Great, thank you all. We have reached our time and gone a little past. We do have questions left, but I will pass those on to our wonderful speakers after this seminar. And with that, I want to thank Alissa, Francis, Ailsa, and Irv for being our speakers today. You said to reach out and contact. What contact information do you have that you want to share? Do you have an email address we can show?
You can either go to the Pacific Marine Mammal Center Sea Lion Cancer Consortium website, or you can email me directly. And my email is, sorry for not putting it up, it's my first initial, so A, and my last name Deming, ADEMING, at pacificmmc.org. And we will be very excited to hear your thoughts.
Great, thank you all. Alissa, I have put that in the chat for people to grab your email if they would like. Thank you all for joining us. I hope you all have a safe and healthy rest of your day or evening. And if you missed this, or if you want to share this with a coworker or a colleague, this was recorded and it will be on the library's YouTube channel shortly. Thank you all.
Thank you for having us.
Thank you very much indeed.
Thank you.